Missing someone. Missing someone. Missing someone.
Hey kids, ever wondered why you're growing?
How does 3 feet pack of flesh turn into a larger pack of flesh? Well, it's all about cell division.
Wait, what?
What? Oh, we meant the reason for cell division. DNA replication.
Look at this. DNA replication is the reason why you look like your parents. They didn't just cut their noses and paste it on your face when you were a child.
First, to understand this, we have to go all the way back to the lab of Watson and Crick. The people who started our suffering, the people who started research about this genetic material.
In this place, they thought about the semi-conservative model of DNA, which means it saves DNA by using other DNA. This was done with an experiment by Michelson and Stroll, but we don't need that, at least right now.
This is by using isotopes of nitrogen, which is something that makes the nitrogen lighter or heavier. At this point, Michelson has proved that the semi-conservative model was correct. Most of you probably got a nosebleed after all that, so let's skip all that first and start with how DNA replicates.
There's an area that marks the start of replication in a strand of DNA called the origin of replication. Kind of like a starting point or origin because it's where DNA began. The DNA replication process is pretty hard to understand, so people studied prokaryotic cells before moving on to this even more complex structure.
Enzymes are key players in this process. Let's start with the helicase. This enzyme unwinds the helical or twisted structure of DNA, like unzipping the zipper of jeans or a skirt. It's formed in the replication fork that has a Y-shaped look. It's called a replication fork since it opens. It looks like a two-pronged fork, or a fork with just two points.
Several proteins and enzymes then prepare the structure for a very lengthy process. There's something called the single-stranded binding proteins to hold the structure in place, to make sure it doesn't coil or go back to its original self. The unzipped DNA is bidirectional, meaning each strand moves in opposite directions. The leading strand going 3' to 5' direction, and the lagging strand heading to 5' to 3' direction.
This means there will be only 2 sides to replicate. But don't worry, the first part is simple. Just the first part. Fear the lagging strand as much as we do.
Don't mind her, she just doesn't like this topic too much. Anyway, the DNA polymerase are the ones that catalyze, or we could say activate, the adding of nucleotides to the daughter strand or the new strand being made.
Things like the clamp loader and beta clamps are used to hold the DNA polymerase in place. They have clamp in their names because they clamp onto the DNA to stabilize the polymerase.
Let's begin with the leading strand, since nucleotides are just added continuously as it goes through the DNA polymerase. It only stops when the DNA is completely copied.
Now the lagging strand. I don't necessarily like talking about this because it's complicated, long, confusing, and an overall pain to explain, and I really wish a Flesh Ranger would just get— oh!
The faster you start talking, the easier we will get over this, okay? Calm yourself. Keep your professionalism.
The lagging strand needs primers to start the process. The primase adds it ahead of the 5' end of the strand. Then the addition of nucleotides can happen part by part, or by segments called the Okazaki fragments. This is done by the DNA polymerase 3, as it slowly adds the nucleotides on. It's a lot, huh?
But moving forward, the DNA polymerase 1 replaces the primers with nucleotides for a smooth and continued strand. Then the DNA ligase makes sure all the segments are bonded and safely together, unlike some relationships in the world.
But finally, once the two are done with their sequences, 2 DNA structures are finally formed. And just like that, it's done.
See? That was easy.
Yeah, sure. Easy. Why do we even have to do this again? Like, why all this hard stuff?
I'm glad you asked. Well, if DNA doesn't replicate, then your cells won't build up. There will be no supply when they do cell division, or when they die. And if that happens, by the time you're 18, you'd be a pile of bones. Not even legal to drink yet.
Okay, so it is important. But I hope that clears things up, kids. The Rafflesia-ravaging scavengers are now out. Yeehaw!
Bye and see you in the next lesson.